All right, thanks, Peter. I want to welcome everyone to today's NCBI Minute. My name is Valerie Schneider, and I'm a staff scientist here at NCBI who leads various teams that are building tools for sequence visualization and analysis. So in today's session, I'll be showing you how to use one of these tools, which is the Variation Viewer, to do interactive visualization of NCBI annotations for variant interpretation. I'm going to be highlighting several tracks in the browser that are useful for variant analysis, and I'll also show you how to visualize your own data in the browser, how to identify and assess variants, and illustrate how the browser integrates with other NCBI resources like ClinVar, PubMed, and dbSNP, where you can continue your variation analyses. I'm going to be using slides today to walk through these various tasks. And the first slide that you just saw in this webinar contains a link to these slides as well as to PDFs and other handouts that cover the same material so that you'll be able to walk through these same steps later at your own pace. So I'll just point out that one of the easiest ways to get to Variation Viewer is at NCBI is via the ClinVar homepage, whose URL is down here at the bottom of the screen. Alternatively, you can go directly to the Variation Viewer browser with the URL that I've got showing down here. So first, let's take a look at some of the features of Variation Viewer that we're going to be using today. In this overview slide here, I've highlighted the two main components of Variation Viewer, the graphical display as well as the variant table below. We'll be using the graphical display to configure the tracks that we want to see of interest, and below this variation table provides information about the variants that are annotated in the region shown here in the graphical display, and the table can be filtered according to a variety of variant characteristics such as the pathogenicity, the allele frequency, and so forth of different variants. And as I briefly mentioned, the table and the graphical display are interactive, such that if you update the region that's shown in the graphical display, the content of the table is going to change accordingly. Other features that we're going to use today are the pick assembly tool, as Variation Viewer supports content from both GRC H37, also known as HG19, as well as GRC H38, also known as HG38. We're going to use the search option to navigate to regions of interest. And we're also going to use the section over here called User Data and Track Hubs in order to add externally sourced data into this browser. So to illustrate the different types of variant analyses that you can do in Variation Viewer, I'm going to use the following hypothetical case study that's described on this slide here. So you can imagine you're working with a collaborator who gives you a VCF file that has 3 suspect variants from a patient who's exhibiting a phenotype suggestive of severe combined immunodeficiency, also known as SCID, suggesting that the ADA or adenosine deaminase gene might be involved. And your collaborator also tells you that this patient additionally suffers from irritable bowel disease. So today, we'll look at how we can use Variation Viewer to evaluate and prioritize these 3 variants that your collaborator gave you, and also to see if we can find any evidence to suggest that the irritable bowel disease is perhaps associated with the SCID-like phenotype. So next, let's go ahead and get Variation Viewer set up for our analyses using the steps that are outlined on this slide here. First, we're going to go ahead and we're going to select GRC H38 for our analyses. As I just mentioned previously, Variation Viewer does support analyses on both assembly versions, but for today, in the interest of time, we're going to take the approach that's described in this case study here, in which our collaborator made their variant calls on GRC H37, but they also provided us with a VCF in which the results were already remapped GRC H38, and which we're going to decide to make use of in order to make sure that we're evaluating the variant data in the context of the updated human reference assembly. So next, we're going to go ahead and we're going to select our tracks, taking advantage of a feature known as the NCBI track sets. And the track sets are pre-selected collections of high-value tracks that are relevant to different types of analyses. So today, we'll use the genetics and variation track set, which contains genes, a track for all SNPs, as well as several variant-associated tracks here, such as SNPs that are associated with ClinVar records, and also SNPs that have publications. Finally, after we've set up our tracks, we're going to go ahead and we're going to upload that VCF file we got from our collaborator. From the options menu here in the User Data and Track Hubs section, I'm going to click the Add Files link, and then I'm going to provide an optional track name, which I'm calling in this case Patient Variants GRC H38. And then once I click Upload, that track is uploaded to the browser, and you can see that the features and the corresponding locations of the variants in that VCF file are now provided in a table in this User Data and Track Hub section. So it's updated to give us this table with the VCF content. Next, I'm just going to go ahead and enter ADA into that search box in order to navigate to that genomic region in the browser, just so that I can get oriented to the display. So right here, I've just shown you that graphical display from Variation Viewer. And what you can see here with this orange bracket are the tracks that came from that genes and variation track set. So at the top here, we've got the NCBI annotation, gene annotation, and we're looking at ADA, where the thin line is the intron, and all these tick marks here are the exons in the gene. Now, down here at the bottom, highlighted with this purple bracket, you can see the VCF-derived track, which we added last. And since I added it last, it's at the bottom of the display. So now I can click and drag on this track to move it directly beneath the genes track to facilitate my analysis in the next few steps. So to navigate to the location of any of the VCF calls in that file that we were provided, we can click on the corresponding row in the table in the User Data and Track Hub section. So we're going to start our analysis today, actually, with the second row, call equals G. So I click on that, and the browser updates to that location. And as I mentioned, our uploaded track has the green highlight shown here, so you can see how I've now dragged it beneath the genes track, as I mentioned before. And the thin line here in the genes track, again, indicates that we're in the intron of an intron in the ADA gene. Now, while most of the other tracks in our track set here are empty, we do see that there is data in the dbSNP all data track. And if we blow this up, we can see that there is a SNP, RS244074, that's annotated at the location of our call. So to learn more about this particular SNP, we're going to make use of the variant table down here at the bottom portion of Variation Viewer. As I mentioned, that table can be filtered by different characteristics. And right now, I'm showing in the table only the SNPs, and I'm excluding any structural variants that might have come from the dbVar database. Now, in agreement with the graphical display that we just looked at, the table tells us that the SNP is intronic, and it also shows us that the 1000 Genomes Project found that the associated A allele at this location has a minor allele frequency of 0. So at this point, we're going to go ahead and remember that our collaborator made their variant call on the GRC H37 assembly. And we also remember that 1000 genome allele frequencies are based on GRC H37. So in this case, the GRC H37 reference allele was an A, and that's the allele that had the allele frequency of 0, which is indicative of a possible assembly error in this earlier version of the human reference genome. Now, I could update Variation Viewer to look specifically at the GRC H37 content to verify the A at this location, but again, that allele frequency suggests that this is an assembly error. So looking more carefully now at GRC H38, we see that the reference assembly now has a G at this position, which indicates that there was an assembly update between the two assembly versions. So based on these data, we can conclude that we're able to actually deprioritize this call from our collaborator because the G that they saw is not actually variant relative to the current reference assembly. So moving on, we can use the user data table again to look at another variant. In this case, we're going to go to the location of call equals A. So we click on that, and this is now the variant that is centered in our graphical display. And if we blow this view up again here, we can see that once again we have a SNP that's annotated at the position of this call. But this time, however, a review of that filtered variant table, where I've got that variant now highlighted in red, shows us that it's an exonic missense variant, and that our call is also a minor allele, which in this case has a non-zero allele frequency as determined by the exact project. So if we click here to expand that row of the table, we actually get additional information about the affected RefSeq transcripts associated with this variant. And we also learn something about the amino acid changes for the variants that fall in exonic regions. Now, in addition to looking at the variant table, we can also notice that there is content in two of our browser tracks, the ClinVar short variations track and the cited variants track. So let's take a look now at what we can learn from these two different tracks. The ClinVar short variations track only includes short variants that are annotated with clinical significance in ClinVar. The color that you see here is used to represent the most severe pathogenicity reported, with the deeper colors corresponding to more severe effects. Hovering over an annotation in this track will bring up this tooltip that provides the variant ID as well as a link to the relevant ClinVar record. If we go ahead and click on that link, we can see that this variation has been submitted multiple times to ClinVar, and it's associated with severe combined immunodeficiency, which is our patient's phenotype. So let's look next at the cited variants track, which contains only variants cited with dbSNP IDs in their publications. The number in this box represents the number of published SNPs overlapping that position. Hovering over any of the features in this track brings up a tooltip that includes links to the corresponding abstracts in PubMed. So if we click on the first of these, we discover that our call or our SNP is located in a hotspot mutation, or is in a hotspot location for ADA. So these data, combined with the data from the ClinVar track and the data that we saw in the table, suggest that this particular call from our collaborator is a promising candidate that we might actually want to pursue further. Now, in order to understand why this location might be a hotspot for ADA mutations, we can investigate the gene annotation at this region in greater detail. So we'll do this by expanding the gene track display, and we can do this by hovering over the track title, which brings up this menu here with this tooltip. And if we click on this cog icon, we get the following menu. And we're going to go ahead and set the display option to show all, and we're going to make sure that we've clicked this other setting to activate product features. So once we do that, our display updates, and we're going to take a fresh look at this gene track. What we see now is our gene features in green. In purple, we have the transcripts, and below in red, we have the CDSs. And beneath this, in black, we have the features that are projected from this gene. In this case, the translations and annotated domains, as well as some other information. Now, if we hover over one of these right here, we find that our mutation, as we can find here with this red arrow, is located actually it's in a metallohydrolase domain, which is very close to the active site residues of the protein. So this information that we're getting from the browser is further confirming for us that this is really a high priority variant and a possible contributor to the observed SCID phenotype in the patient. So let's move on now using the user data table and navigate to our third candidate, call equals T. So once again, our display is updated to put our call in the center of the graphical display, and we can see that once again we have another intronic variant. And while once again there is a known SNP annotated at this location, it turns out that there's no data in the ClinVar cited variant or any other track in this genes and variation track set to suggest any relevance of this call to the SCID-like phenotype in our collaborator's patient. Fortunately, NCBI has many additional tracks available. So in order to further investigate this call, we're now going to make use of a new track that's available from RefSeq at NCBI, known as the biological regions track. So features in this biological regions track include non-genic functional elements that have been experimentally validated. So in order to get this new track into the display, we're going to click on this tracks button that's up here at the top right, and we're going to select configure tracks. In the resulting dialog that opens, we're going to go ahead and click on the search tab, and then we're just going to start typing in the search box biological regions, and we'll get some results back, and we're going to go ahead and select the first result right here, which corresponds to the functional elements that were included in release 109, which is the latest RefSeq annotation of GRC H38. So then when we go ahead and click configure, this track is going to get added to the display. Now, for ease of viewing today, I've gone ahead and already dragged this track up towards the top of the display, just above our patient call track. So here's our call, and here in aqua and red are the biological region features. What we can now see is that our call is within sequence that's been annotated as functionally relevant. Hovering over any of these features brings up tooltips with information. In this case, we see that our call overlaps both an enhancer and a locus control region in ADA that control expression in the thymus. The tooltips also include links to the relevant abstracts in PubMed where this experimental evidence came from for this biological feature. So from these results, we're able to conclude that our variant that our collaborator gave us could potentially have a functional effect on ADA expression despite being located in an intron. Thus, we'd also want to go ahead and prioritize this variant for further review. So this is information we can feed back to our collaborator. Lastly, let's return now to that irritable bowel disease phenotype that our collaborator mentioned to us. The genetics and variation track set that we've been using also includes a track called GAP Plus NHGRI GWAS Catalog and dbGAP Submitted Association Analysis results, which features the top GWAS results submitted to dbGAP plus published results from the NHGRI GWAS catalog. I'm using this slider up here at the top of the graphical display now in order to zoom in order to get a better, to zoom out, in order to get a look at whether there are any results in this track that are near the ADA gene. So I'm going to use the slider to slide back to this zoomed out position here. And when the display updates, in this view here, I've just highlighted for you here in red the position of the ADA gene, and down here are those 3 calls that our collaborator gave us. And down here at the bottom now is that NHGRI GWAS catalog track. And you can see that there are actually some hits in this track that are just downstream of the ADA gene. So blowing this up, we can take a better look at the content of this particular track. In this track, both the scatter plot above these bins and the bins themselves reflect individual p-values. So both the color and the dot's location correspond to the p-value, and we're also getting here the count of reported associations for that SNP. So if we hover our mouse over the bin that has the top hit right here, we get a tooltip that opens up, and we see that this particular SNP, which I've highlighted here in red, is a result from the GWAS catalog, and it reflects an association with ulcerative colitis. And there's a link down here to PubMed so we can get more information. So by looking at the content in this tooltip and this track, we're able to tell our collaborator that they might want to follow up on this particular information to further evaluate a putative link between ADA, and irritable bowel disease. So while this concludes the variant analyses that we're going to do today in our webinar, I also want to point out that there are other browser features that you might find useful in your analyses. First, I want to emphasize that you can share your configured display with your colleagues by clicking on this Share This Page link at the top right of Variation Viewer. Secondly, you can save your track configurations in the display to your NCBI account using the NCBI Track Collections option. It's here underneath the Tracks menu. Lastly, by clicking on the Tools menu, you can go ahead and generate printer or publication quality PDFs of the display. And these are vector graphic images that you can then update to further modify as you need. So with that, we've come to the end of our case file and our analyses. To look at 3 variant calls in the ADA gene. Using the browser, we were able to discount one of these calls as an artifact of GRCH37 assembly error. And we determined that the other 2 are candidate SNPs, one being an exonic missense variant and the other being an intronic variant. We also used the integration of Variation Viewer with ClinVar and PubMed to get additional information about one of the variants. And we used the new biological regions track to uncover a potential functional implication for this intronic variant. Lastly, we used the GAP plus GWAS track to look for studies that might be associated with a possible secondary irritable bowel disease phenotype. So you can use these slides, the PDF, and the corresponding YouTube video that you'll be able to pick up after this webinar to walk through these steps at your own pace. We hope you found this NCBI Minute informative, and please let us know if you have any additional questions about the browser or the data in the tracks. So thank you.
Okay, thank you, Valerie. Okay, well, there don't be any questions right now, so I'll go ahead and end the webinar, and we'll talk to you again soon. Thank you for coming.